right, now I would like to invite Luis Williams, who is with Quiver, to talk about their ASO work on STXBP1.
Thanks, Charlene. So I'd like to, again, thank the organizers of the meeting for inviting me to present during the research meeting and also during the family meeting. So I was tasked to give you a lightning-style talk with some basic principles and concepts related to antisense oligonucleotides as a therapeutic modality for STXBP1 and other CNS genetic disorders. So I work in Quiver Bioscience, as Charlene mentioned, head of discovery. My training is as a scientist in stem cell biology. So I spent a few years learning how to reprogram cells, how to turn the stem cells into neurons to try to understand what's going on in several genetic disorders of the nervous system, and then use those models to both create a mechanistic hypothesis and also discover potential therapeutics. So before I tell you what ASOs are and how they work, I think it would be good to revisit a little bit the flow of genetic information. I've seen a couple of talks throughout the day that have shown this. So I just want to highlight a couple of things, right? As it was mentioned earlier, right, when we think to the end stage here, protein, right, so all of those green dots, right, those proteins represent the machinery of the cell that carries out a lot of function. So STXBP1, as we just saw in the previous talk, is a protein located in the presynaptic terminal and is involved in how the cells talk to one another. For the cells to make a protein, right, those instructions come from the nucleus, from the DNA. And you can think of the DNA as really a large collection of instructions in the form of letters. And there's only 4 types of letters, but there's actually every cell contained in our body 12 billion letters. That's a large amount of information. Now, when a cell is about to make a protein, right, there is a very, very tiny fraction of that entire collection of letters that gets read, and that process is called transcription, and you generate this intermediate molecule called pre-mRNA. Now, by the time you get to that intermediate molecule, now we're talking about a few tens of thousands of letters. That pre-mRNA gets further modified in the nucleus, gets shortened, and you generate a very concise message, and that's called messenger RNA. And that is now a few thousand letters. For instance, STXBP1, it's around 3,800 letters, right? So that's how you get from DNA instructions all the way to protein. So what is it that the ASOs do? So I was trying to come up with a metaphor to explain this process, right? And I came up with something that sort of works, but you have to forget about computers and smartphones for a second. So imagine, right, that the protein is a meal that you have to prepare every day on a daily basis. To prepare that meal, you need a recipe, right? But to get that recipe, you can only get it from this large book that you have in the office. That's the nucleus. And you have this massive book that has thousands of recipes and is so heavy and it cannot be digitized. And the only way for you to get the page with the recipe is to, make a photocopy of it, right? That's why I was saying it's an analog metaphor. So you make a photocopy. Now you have that page. That's the initial recipe. That's the pre-mRNA, right? You can then, before you go to the kitchen, make a few revisions to that recipe. You can scratch a few ingredients, you can shorten it, and that's the recipe that then you take to the kitchen and make the protein. You make the meal, right? And then every day you have to do that because let's say the photocopy fades and you have to go back to the office and do this. So when we think about ASOs, what ASOs are doing, they're actually, there's genetic type molecules. They're actually made of the same type of letters that make DNA and RNA, but they're very small, right? Instead of being tens of thousands of letters, there's only 18 to 20 letters. And what they do, in essence, is they bind to either the recipe or the revised recipe. And depending on the chemistry you're using, you use the ASOs to modify the amount of dish that you're making. Are you making a lot? Are you making more? Are you making less? So this is now a cartoon of that interaction, right, of the ASO molecule, and each of the little bars is a letter. So the ASO, it's about 18 to 20 letters, and this is a very small representation of a piece of the pre-mRNA, right, the photocopy recipe, or the mRNA, the final recipe. So like I said, depending on what you do to that ASO, the type of chemistry you're using, you can use that to have different outcomes in the cells. You can use an ASO to degrade the final recipe. So you can use the ASO to make less of a protein, and that's what's represented in module A. You can also use the ASO to bind to just the photocopy recipe, the first version, and have an influence, right, on the amount of ingredients and how the final recipe gets put together to try to increase the amount of the dish that you're making. You can also have the ASO bind the final recipe and stabilize it, right? So I mentioned about the photocopy, being faded after 24 hours. You can make it last longer, so that way you can use it in the kitchen multiple times and also increase the amount of dish that you're making. So you can imagine, right, that depending on the therapeutic disorder that you're targeting, you're either going to go with destroying the recipe or changing the recipe to make more of the dish. So I'll give you a couple of examples of those two approaches. Now, as mentioned before, currently there is no way to target ASOs, right, to the brain and spinal cord. You cannot do an infusion of ASOs and have them cross the blood-brain barrier and get to the brain and spinal cord. But these molecules can be delivered and are delivered intrathecally. So there's a lumbar puncture. Patients get treated with ASO 3 or 4 times a year. Now, the advantage of that, of course, with having multiple opportunities for dosing, is that you can either escalate, right, if you see hints of efficacy and want to do more, or if you see some hints of adverse events and you want to try to control for that, you can lower the dose. Right, so that's some of the advantages of the multi-treatment, but it's it's an intrathecal delivery, so it's not a neutral intervention. So a couple of examples of ASO therapies for genetic disorders of the nervous system that are in the market. So the first one, which was approved in 2016, was for spinal muscular atrophy. So this is a motor neuron disorder in which the spinal motor neurons, these are the motor neurons in the spinal cord, they die because they don't have enough of this protein called SMN. This is survival of motor neuron protein. So as I mentioned, right, that you can use a strategy with an ASO to modify the recipe to try to make more of that protein, and that's what's being delivered to the patient, and it's worked successfully. There's more than 11,000 patients that have now been treated with this ASO therapeutic. The other approved drug of ASO class in the market, which was approved 3 years ago, this is for another motor neuron disorder. It's called ALS. So it's motor neuron death in adult patients. And this is to treat a particular form of ALS that is caused by mutations in this gene called SOD1. So you're making a toxic protein that aggregates. So you can reduce the amount of that protein, right, by modifying an ASO that will go and will degrade the recipe, so you're making less of that protein. Now, for STXBP1, we're using module C, right, at Quiver. We have designed ASOs that will bind to a particular region of the recipe where we think we're stabilizing the recipe to make more STXBP1. As it was described very, very nicely in the previous talk, right, STXBP1 is a haploinsufficient disorder. You have, patients have a defective copy. One of the 2 copies is defective, so you're making close to 50% of the protein, or the protein you're making has reduced function. To do the assays, you have to be able to measure protein, and that's what those 2 dark bands are showing. The one on the left and the upper set of bands, right, is the 2 wild-type copies of STXBP1. The one on the right has 1 defective copy, and you can see the difference in intensity. So what we want to do is design ASOs that we can treat the heterozygous neurons and then see an increase of STXBP1 level. So that's what we have done at Quiver. We have done 2 rounds of design and screening. In the first wave of design, we tested around 35 different molecules in multiple rounds in different neuronal lines and identified 2 different ASOs that could increase STXBP1 protein levels at least 1.5x. And what is showing on the left is the actual assay. We have the stem cell-derived neurons. We treat them with the ASOs, and 6 days after, we're collecting the protein and measuring STXBP1 levels. And you see the ASO mark with a star, ASO 29, is giving you that thicker band, right? So that's higher levels of STXBP1. And you see, for instance, that's in comparison to the vehicle. That means just treating the cells with a buffer, no ASO. Or you can see that in comparison to ASO 13. So that would be an ASO that did not work in increasing STXBP1. Now, we have done a second wave of design and screening, and we have identified 15 additional molecules that work. We have tested these molecules in just 1 round. Now, these molecules, this last set of 15 molecules, have full homology with non-human primates. The reason that's important is because at some stage in the preclinical path towards FDA, we'll have to test these molecules in non-human primate. And they're more likely to work if they have full homology to the STXBP1 version of in monkeys. So this is the last slide, just to show you a little bit what are some of the next steps, and to compare those with 2 of our other preclinical stage programs that are a little more advanced. So for STXBP1, right, so we have a set of molecules that we're further characterizing, and we want to build in vitro efficacy assays, right? So in addition to proving that the molecules can boost STXBP1 protein levels, you want to demonstrate, similar to some of the data that was being shown a couple of talks ago, that when you treat the neurons, you're changing the neuronal behavior of neurons that have an STXBP1 deficiency. After that, right, the next set of modules focus on safety tolerability, right? These molecules will go to the central nervous system, so you want to pressure test those molecules in both mouse and rat. So you inject them at a very, very high concentration in the central nervous system of these animals, both ICV in the case of mice and intrathecal in the case of rats, to demonstrate that they don't have any toxicity effect, right? They're not changing the functional behavior of those animals after, up to 60 days after injection. So you do histopathology as well. After that, and I'll just mention this follow-up study, right? You want to have at least 2 or 3 molecules to initiate your first non-human primate study. So for that study, right, you want to have molecules that you're very, very confident are going to be safe in the non-human primate and are also going to give you target engagement, so efficacy, right? So that goes back to the importance of having molecules that are full homologous to the STXBP1 in monkeys. So we have a couple of preclinical stage programs at Quiver. One of them is targeting a sodium channel for chronic pain. For that one, we just finished the first monkey study. We have a candidate and a backup that we're testing now in additional monkey studies. And our next program is in DUP15Q syndrome. So here we get duplication of a genetic region of chromosome 15, and we're targeting the reduction of UB3A. For that one, we're about to initiate the first non-human primate study next month. So we're hoping to replicate that path of our first 2 program with STXBP1 this year. So with that, I want to thank especially the STXBP1 Foundation. I want to thank all of you for staying till this talk. Again, this type of meeting is really a driver of motivation and inspiration to continue doing the work we do. So thank you so much. And I'm happy if there is time to answer questions, or I'm staying as well. I think my flight just got canceled. So feel free to come up one-on-one if you want to talk ASOs, neurons, stem cells, et cetera. So thank you so much.
Sounds like drinks at the bar. Does anybody have any questions right now for Luis? Great. Oh.
How are you funded?
How are we funded, right? So we have we're actually raising Series A right now to keep to advance the first program to the clinic. For our STXBP1 and the DUP15Q program that we have done, a lot of the early discovery work was done with angel investor. We've had a few grants, and there's a couple of investors for the DUP15Q program that have funded the non-human primate study that's about to initiate as well, and also through grants with the DUP15Q Foundation.
Thank you.
Mm-hmm.
And you can find Luis later today.
Thank you.